3 years ago, this animal was discovered, Tiktaalik. It was being sought intentionally in sediments of 380 million years of age in northern Canada, in the Inuit territory, because these other two animals were known. This starts to show some signs of being an amphibian, but this is still by and large a fish. This is clearly an amphibian, and as I said, 3 years ago, Tiktaalik was discovered, clearly intermediate. But what concerned mostly the contemporaries of Darwin and other people who asked the question, where are the intermediates? Are the intermediates between the common ancestor of our closest relatives, the chimpanzees, and humans? By the time Darwin died, none of these intermediates was known. 7 years later, in 1889, the first one was discovered, something now called, a creature now known as Homo erectus. It was discovered by a Dutch physician in Java, in what is today Java, and he called it Pithecanthropus erectus, Pithe and Anthropos, mono-human erectus, had a small brain, but clearly this physician who knew anatomy was aware that this individual had to have walk on two legs, that is, that had an erect gait. Since then, thousands of intermediates of these hominids, which are the intermediates between our ape ancestors and modern humans, thousands of these have been discovered. Literally, dozens of them new ones are discovered every year. An important one that I am going to use to illustrate a principle is Australopithecus afarensis, Lucy. The point that I want to make is, according to the theory of natural selection, evolution will not be something that happens gradually in all respects. That's the way his contemporaries thought evolution happened. Darwin said, no, different parts will evolve at different times in response to the needs of the environment. And here you have a classic example. If you look at two of the major features that distinguish us from the apes, one is we have a larger brain, 3 to 4 times larger than that of a male chimpanzee, and then the bipedal gait. And if we look at Lucy, Australopithecus afarensis, here is what was found 40 years ago, 40% of the skeleton of an individual. But I want you to look at the hip bone, the pelvis. Good anatomists will have many other ways to find out that Lucy had bipedal gait. But if we look at the pelvis, I have enlarged it because Lucy could have been a 3 feet tall young woman. I have enlarged it in size to compare the shape with that of the modern human and with that of a gorilla. And it's clearly that Lucy could not walk the way a gorilla or chimp walks, knuckle walking, resting on the knuckles, because the hip bone was not large enough, so he had to walk like that. But the brain will only become larger much later, starting about 2 million years ago. Darwin used also comparative anatomy, and here I'm using a simple example: the bones of a dog, a bird, a whale, and a human. The 4 limbs are made of the same bones, organized in the same way, and yet they are used for completely different purposes: for swimming, for flying, for running, and for handling objects or for writing. And the argument of Darwin was, how do we explain that similarity? An engineer does not design an airplane and a ship and a car with the same parts organized in the same way, but uses new materials for each purpose. How do we explain this similarity, he says, from a common ancestor, which had already this structure in the forelimbs, actually from Tiktaalik, although he didn't know about Tiktaalik? Well, the most convincing evidence for evolution comes from a scientific discipline that don't exist in Darwin's time, and not for a century since the origin of a species, molecular biology. Now we know the genetic information and the evolutionary information is encased in the DNA, in the succession of these letters, and a human genome has 3 billion of these letters. They have evolutionary and genetic information. If we were to write all the letters of a human genome, we will need 500 volumes of the size of the Bible. That's how much genetic information we have. And every little part of that information we can use to reconstruct evolutionary history. By the way, I carry my DNA on my necktie. I don't know if you can see it. This was done copying from a book of mine, that model. The first demonstration of this was done in 1967 with a very small molecule, because at the time we still could not handle full genomes, not even large genes as we do now, large proteins. This is a very small protein, and it was done by two scientists. The leader was Professor Walter Fitch, who was at the time a very young scientist. Now he's a senior professor in my department at the University of California in Irvine. But this was in 1967, and this is how molecular evolution works, the comparative study to find out the relationship between different organisms. Here you have humans, monkeys, and horse, and here you have the sequence of amino acids as they go, so that you align them. And when you align them, you find they are mostly similar, but there is one difference only between humans and monkeys, and 11 of 12 between humans and horses.
It's getting to be time to wrap up.
Sorry?
Time to wrap up.
Okay, I will finish in 1 minute or two. So here we have the comparison in a simple matrix. Now, they did a comparison between 22 organisms. You have this matrix. You feed this matrix into a computer, given the computer the two basic laws of evolution, and the computer produced this. An evolution which covers 2 billion years. One branch goes to the yeast and fungi, another branch goes to insects, another branch to the vertebrates. Amazing, a little molecule. Now we can use thousands of genes to do this, and indeed we have reconstructed the whole evolution of all organisms in the world by these methods. Thank you. Thank you.